In brains, there are all of these neurons, and they make connections with each other. So they talk to each other, communicate with each other, and the really strong idea, strong hypothesis, is that everything in a brain, all of the memories of that brain, the personality of that brain, the skills, fears, etc., is in the map of those connections. So that if you knew the map of those connections, you could figure out all these other things about the brain. And so, to what extent does mapping symptoms to brain networks with the connectome open windows of understanding into the brain? The brain is something we as humans have always strived to understand, but just looking at someone's head won't magically unlock all the secrets kept within. And so, neurologists and radiologists have spent decades working on a piece of technology that is slowly unlocking the once secrets we so desperately desired. This is the human connectome. The human connectome and its medical purpose. The human connectome is a wiring diagram of the human brain derived from specialized MRI scans across thousands of individuals. According to Dr. Rafael Rojas, this wiring diagram is formed through the extensive process of utilizing specific MRI scanning techniques such as EP2D DSI T1 weighted multi-echo MP-RAGE, which is the process of brain morphometry forming a detailed three-dimensional image of the brain. The specialized MRI scanners used can analyze a water molecule's movement in the brain by a thousandth of a millimeter. Additionally, the scanner acquires the raw data of the brain, which is approximately 100 megabits of data, though, once fed through mathematical models, the output of the 3D image can reach sizes of 2 million gigabytes of data, compared to the human genome, which in full was only 3.3 gigabytes. The acquisition of the brain's neurological pathways can assist in the identification of lesions or areas of dysfunction. The purpose behind the human connectome is to form a generic map of the human brain which can be used to compare with people who have neurological disorders and diseases. This process intends on aiding the diagnostic process of neurological issue causal factors. MRI or magnetic resonance imaging utilizes natural magnetic features of the human body to produce intricate images. In the MRI process, a hydrogen nucleus is used as it is prominent in both water and fat within the human body. An MRI machine acts as a strong magnetic field, and such, when a body is placed within, the proton's axes within the hydrogen nucleus align. Radio waves can then be added as an additional energy source to deflecting magnetic vector formed from the proton's alignment. The frequency present in these radio waves cause the resonance of the hydrogen nuclei, and hence, through the alteration of gradient coils, different parts of the body will resonate. The final image is formed once the radio waves are turned off, and the vector returns to resting state. This process causes the emission of radio waves, which, once identified, can be used to form an MRI image to be utilized in the human connectome. According to Dr. Michael D. Fox, the human connectome's primary focus is on mapping the anatomical and functional connectivity within a healthy human brain. By doing so, the future purpose is to utilize such data and facilitate research in regard to neurological disorders such as schizophrenia, Alzheimer's, dyslexia, and anxiety. Mapping symptoms to brain networks. Rather than try and identify a single brain region causing a symptom, neurologists are attempting to identify the specific brain network or brain circuit causing a symptom. Lesions or symptoms can be mapped onto specific brain networks with the aid of connectome scans. By utilizing the network localization approach to determine a concise and consistent neuroanatomical basis, immensely diverse brain injuries in regard to neurological locations can have its lesions placed on one of two distinctly identifiable brain networks. According to Dr. Wanyan Lim, to map symptoms to brain networks, numerous variations of imaging must be utilized. Firstly, diffusion imaging or DMRI scans collect a series of 3D volumes. These volumes, or B0 images, have diffusion gradients applied to form a viewable image. This specific scan is ideal in the identifying and characterizing of axons and their spatial arrangements in the white matter segment of the brain. The second is tractography-based imaging of brain pathways. This process of tractography algorithm focuses on stimulating water molecules movement in the brain's axons to form path-like images. These two key methods of formulating images are then enhanced with the application of the connectome imaging and how mapping brain symptoms can be utilized to conduct research into neurological disorders. With the high resolution data which is collected from connectome imaging, a detailed pathway of the human brain can be reconstructed with completeness. This process also captures the considered more fine-grained details of the brain. The idea of free will has been identified as the combination of two key processes in the cognitive brain. These are the desire or want to act, known as volition, and the understanding and sense of responsibility for these actions, known as agency. From this, it has been identified that lesions to areas of volition and/or agency in the human brain can leave patients without the ability to want to move, speak, and realization that their movements are their own. This discovery was made by utilizing the process of identifying brain lesions and mapping their characteristics to specific brain networks with deep analysis to determine the anatomical origins of their symptoms. This is just one prime example of symptom mapping in the real world. Lesions and their connection to neurological disorders. A lesion is an area of organ or tissue that has undergone a level of damage. This can be caused by injury like concussion or disease like ulcer, tumor or abscess. In this process, lesions are used to localize neurological symptoms. An example of this is when someone has a stroke. A symptom, for example paralysis, can be connected back to the area of stroke which has formed such lesion. This lesion analysis, when coupled with the human connectome, allows specialists to determine whether or not the lesions in different areas or sites can cause similar, if not the same, symptoms. This is done by taking the lesion's location and applying it to the human connectome, and then analyzing the brain networks in such area and their effect on the human cognitive and psychiatric systems. A connection to neurological disorders can be identified when using lesion network mapping and applying it to a variety of neuropsychiatric disorders. An example of a lesion and its connection to a neurological disorder is cerebral infarction. Cerebral infarction refers to the death of a tissue. This type of lesion is also commonly called a stroke, when a cluster of brain cells die due to a lack of oxygen and blood supply. These types of brain lesions are diagnosed through the process of CT and MRI imaging. By doing so, the characteristics of the lesion can be identified and attempted to be repaired. Brain lesions are treated in numerous ways dependent purely on the type, severity, and risk of death. Common approaches for treatment are as followed: surgical removal, chemotherapy and radiation therapy if cancerous, medication such as antibiotics if infected, anti-inflammatory medication, and immune system calm. According to Dr. Donald D. Burrow, by doing so, localization lesion analysis has allowed for symptoms like auditory, criminality, Parkinsonism, and coma to be identified. In specific, lesions that were spread across the brain in different locations caused the same symptoms. For example, Parkinsonism, due to the lesions aligning with similar brain networks, though placed in different locations, caused the same symptoms. These results from work with lesion network mapping have given insight into neurological and neuropsychiatric disorders, and hence allowed for accurate diagnosis and, in specific cases, treatment and cure. How this process has opened windows into the understanding of the brain. A vast majority of symptoms do not map to specific brain regions, but do map to specific brain networks. This has allowed for complicated symptoms to be mapped, such as consciousness, criminality, and disorders of free will that were previously not possible. The process of lesion network mapping has seen numerous clinical applications and has many aims in regard to future directions. As a tangible measure, network mapping has led to the effective treatment protocols. For example, patients who suffered from extensive hallucinations found to have extrastriate visual cortex, which was targeted with transcranial magnetic stimulation. Dr. Michael D. Fox, in an interview with the New England Journal of Medicine, explored the future endeavors and possibilities of the Connectome project.
That there are a lot of basic science applications of having a wiring diagram of the human brain. I think that where it's going is we're going to be able to map each individual symptom to a specific circuit. Maybe not a complex disorder like schizophrenia or autism, but symptoms of schizophrenia and autism will map to specific circuits and will come in with therapies specific to those circuits. It's going to come down to treating people's individual symptoms, not their diagnosis, by targeting individual circuits that underlie those symptoms.
Schizophrenia is a severe psychiatric condition encompassing hallucinations, loss of initiative, loss of cognitive function, and delusions. As the causes of schizophrenia are still unknown, it is hypothesized that it occurs as a result from abnormal anatomical neuroconnectivity and a consequent decoupling of the brain's integrative thought processes. Advances in high field structure and functional neuroimaging techniques have allowed for the reconstruct of macroscopic neural wiring systems of the human brain, known as the connectome. By utilizing the connectome's accuracy, depth, and advanced neural wiring capabilities, areas of the brain which see little activity and/or perish can be connected to specific brain networks and their functions. By doing so with substantial evidence, repetition of study, and correct circumstances, the causal factors of schizophrenia will soon be understandable. This is just one of the many advances the connectome scanning process has made in the understanding of the brain. The human connectome is a revolutionary development in the region of brain understanding and anatomy. Aided with specialist symptom mapping techniques, has the potential to discover causes of neurological disease and disorders which are currently unknown.
We want to understand and be able to ask deeper questions about behaviors and where they come from and why am I different from you and why are we similar. So it's a very exciting time in developmental neuroscience.
And so, with the constant development and continuous research into understanding the brain, the human connectome will play a vital part in conjunction with mapping symptoms to brain networks to open windows of understanding into the brain. It is essential that symptom mapping with the connectome is endorsed, as it is paving the way into the future of alleviating, understanding, and curing neurological disease and disorder.